Hello, hello everyone.
Good evening, ma'am.
Good evening, Shreya. How are you?
Fine, ma'am.
Where is Risha today? Anavadya? Risha and Asmi?
Mama, I don't know. They probably joined.
No, no, I can't see them. My camera is not working properly today, so I'll be able to like
Uh,
on my video.
Ma'am,
Ma'am,
Yeah.
even mine is not working.
Okay.
Asmi said that she will join like 10 to 15 minutes late.
Okay. And what about Risha?
Ma'am, Risha, she didn't message me.
Okay, just a minute. Let me see. Go and get my mobile. Someone messaged me right now, but I have not seen that message. So it might be possible that uh, go bus, stop. Wire is there. Go out. Thank you. Risha. Okay, she will also be joining 15 minutes late for the class. Okay, then we can also do one thing. We can also start class after 10 minutes because, or should I start class? I think I should start. Otherwise, our time will be wasted and Shreya will have to leave after 5, है ना? Right, Shreya?
Yes, ma'am.
Okay, so I'll start right now. So, have you finished reading that chapter number 18? Did you find any problem in that? The theoretical portion? Both Shreya and Anavadya.
Ma'am, I didn't get time to read. I was writing my holiday homework. I'll read it today, ma'am. I'll tell you
Okay.
tomorrow.
Okay, it's fine. Read it and ask me if you have any question or any problem.
Yes, ma'am.
Anavadya, do you have any problem, Anavadya?
No, ma'am, no, ma'am.
Okay. Did you studied that? Have you gone through
Yeah,
that?
yes, ma'am. I read it, and we also had like class at school today with the same topic. So yeah, I didn't
Okay,
find
okay.
any difficulty.
Okay, fine. So today I'm going to start this chapter, what named chapter number 19. Okay. So, uh, are you able to see my screen?
Yes, ma'am.
But I am not able to see. Wait. Can you see chapter 19 notes? It is visible to you all?
Yes, ma'am. It's visible.
Okay, now please on your video so that I can see your face at least. Okay, only Anvadya's camera is working, right? Shreya's camera is also not working. Mine is also not working. I don't know what happened today. Okay, so okay. So today we are going to start chapter number 19, that is excretory products and their elimination. This is very interesting chapter and very easy too. Okay. So like in animals, ammonia, urea, uric acid, carbon dioxide, water and many ions like sodium, potassium, chlorine, phosphate, sulfate, etc. They either they are uh, formed as a result of metabolic activities that are occurring in us or by many other means, like if we ingest excess amount, then these we can say what they are few ions and something that are formed in our body. So it is very much necessary for our body to remove them totally or partially, otherwise they may be harmful for our body. So the mechanism that involves the elimination of these substances that are produced in our body, and they are mainly nitrogenous waste. So how these nitrogenous waste are eliminated out of our body is will be explained in this chapter basically. So this ammonia, urea and uric acid, they are the major form of the nitrogenous waste that are excreted out by the animals from their body, out of which ammonia is the most toxic form, and it requires a large amount of water for the elimination of ammonia from our body, while uric acid is the least toxic and it can be removed with a less amount of water, and the process of excretion of ammonia is known as ammonotelism. So if someone asks you, like, out of ammonia, urea and uric acid, which one is the most toxic, then you should reply ammonia. Okay, Shreya,
Yes, ma'am.
are you hearing me, beta?
Yes, ma'am. मैम वो
Okay,
बीच बीच में ब्रेक हो रहा है
my
नेटवर्क
voice is
कनेक्शन
my
मेरा
voice is
नेटवर्क मैम मेरा नेटवर्क ठीक नहीं है
ओके 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 बेटा, कोई बात नहीं. आई थिंक आज टिल टुडे नाइट आई विल बी सेंडिंग यू द नोट्स ऑफ दिस चैप्टर. I will be sending you all, so
ओके मैम
you can read and come in class before like our class will be started.
ठीक है मैम
Fine, Anavadya?
Yes, ma'am.
Okay, beta. And I'm also planning to take test of that chapter. Which chapter? Chapter number 17, I think. 17.
Breathing.
Last two chapters we have done 17 and 18, right?
Yes, ma'am. Yeah.
Okay. So I'll discuss with you all and then we'll be taking test for that chapter. I think I should take test after your assessment will be submitted, right? Otherwise it would be burden on you. You are writing biology assessment also, right?
Yes, ma'am. Yeah.
So you will be submitting it till 7th, okay?
Yeah.
So I will be taking test after that. Fine? Okay. Okay. So in case of many bony fishes, aquatic amphibians and aquatic insects, the thing that is released out of the body is ammonia, hence they are known as ammonotelic. Okay. So you should know the difference between ammonotelic, ureotelic and uricotelic. Okay. So those animals in which the ammonia is released in form of ammonia is released in form of nitrogenous waste is released in form of ammonia, then they are known as ammonotelic, and the examples are given here. And uh, terrestrial ammonia is readily soluble in water, and it can be generally excreted out by the process of diffusion across the body surfaces in case of these aquatic amphibians with the help of gills in case of ammonia in form of ammonium ions, and in case of fishes kidneys do not play a very important role in removal of this nitrogenous waste. So if it is asked, like in case of fishes ammonia is released out of their body, then which organ is responsible for excreting out ammonia? So you can say it is excreted out by the process of diffusion across their body surface or through their gill surfaces. Okay, in case of fishes ammonia is released in form of ammonium ion through their body surface or through their gill surfaces. So kidneys are not playing a very important role in case of fishes for removal of ammonium. Okay, it is an exception, otherwise filtration and this release of urea and uric acid always occur with the help of kidneys because they are excretory organs, but in case of fishes kidneys are not playing this role. Okay, so the terrestrial adaptation it make it necessary for production of lesser toxic nitrogenous waste like urea and uric acid. Out of urea and uric acid is the least toxic. Okay, is the least toxic and ammonia is the highest toxic. Okay, and why it is done so? It is done to conserve the water. Otherwise ammonia, the release of ammonia from body requires a large amount of water as well. So if we require such a more amount of water for ammonia to release, then there will be deficiency of water. So in order to conserve the water, the terrestrial adaptation makes it necessary or it is required that we should produce lesser toxic nitrogenous wastes like urea and uric acid that require less amount of water for release of these toxic materials out of the body. Okay, fine. Shreya and Anvadia, should I proceed, beta?
Yes, ma'am.
Okay, is there any doubt till now?
No, ma'am.
Okay, so a monotelic I told you earlier. Now I will tell you about uricotelic. What are uricotelic? The mammals and many terrestrial amphibians and the marine fishes. See, it is not necessary that all the fishes are aminotelic. Which are aminotelic? Just a minute. Uric acid. Okay, so bony fishes will be aminotelic and marine fishes will be uricotelic. See the difference, don't get confused. If it is asked that these bony fishes, they will be aminotelic, whereas the marine fishes will be uricotelic. Sorry, ureotelic. Okay. Mammals and many terrestrial amphibians and marine fishes mainly excrete urea and are known as uricotelic animals. So those animals that releases or discharge ammonia are known as ammonotelic, and those animals which releases or excrete out urea as the nitrogenous waste in form of nitrogenous waste, then they are known as ureotelic. Okay, so difference between aminotelic and ureotelic is clear along with the examples. Anvadya and Shreya, should I proceed, beta, or is there any doubt? These are just simply the like definitions. What are ureotelic and uricotelic and aminotelic along with their examples. So you should learn at least these examples. Fine, Shreya, should I proceed?
Yes, ma'am.
Anvadya, okay?
Yes, ma'am.
Okay. So ammonia produced by metabolism. What happens after the ammonia is produced in by different metabolic activities in our body? So the ammonia that is produced by metabolism is converted into urea in the liver of these animals and released out of the body, released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may also be retained within the kidney matrix of some of these animals to maintain the desired osmolarity. Okay. So this ammonia that is produced in the body is converted into the urea within the kidneys. Okay, then this urea is released into the blood and then again it is filtered and excreted out with the help of kidneys. Okay. So how the urea is formed? It is formed from ammonia only. Lekin it is produced like converted into urea within the liver. Don't forget, remember this thing that the ammonia that is produced in our body by different metabolic activities is converted into the urea, that is the less toxic form. So this conversion of ammonia into urea will occur in the liver of the animals and then this urea is released into the blood, that is then filtered and excreted out by the kidneys. Okay. So reptiles, birds, land snails and insects, they excrete nitrogenous waste such as uric acid in the form of pellets or paste with a minimum loss of water and are known as uricotelic animals. So ureotelic, aminotelic and uricotelic. The difference is clear between these three. Disha, please on your video. My camera is not working today, so I will be not able to on my video today. Okay.
Yes, ma'am.
Okay. So till now I have told them what is the difference of ureotelic, uricotelic and aminotelic. Okay. So the animals that excrete out the nitrogenous waste in form of ammonia are known as ammonotelic. And the examples of ammonotelic are uh, many bony fishes, aquatic amphibians and aquatic insects. And the ammonia is the nitrogenous waste that is the most toxic and it requires a large amount of water for its discharge or like removal from the body. And the animals that releases urea in form of nitrogenous waste are known as ureotelic. And examples of mammals, many terrestrial amphibians and marine fishes. And the animals that excrete nitrogenous waste in form of uric acid, and this uric acid is released in form of pellets or paste, and it requires the minimum amount of water for its discharge, and they are known as uricotalic. And the examples are reptiles, birds, and land snails. Okay. So this was the division for you, Disha. Till now, I have taught them this much. Okay.
Yes, ma'am.
Okay. So like in most of the invertebrates, different excretory structures are present in different animals. In case of most of the vertebrates, the structures that are responsible for excretion of nitrogenous waste are simply tubular, while in case of vertebrates, they consist of complex tubular organs known as kidney. And this thing I have already taught you in some of the chapter. I don't remember the name of the chapter, but these different excretory structures, I think I have taught you in earlier class also. I don't remember in which chapter, but again I'm like repeating the same. Okay, so some of the different type of excretory structures present in different animals are mentioned here. Hello, Asmi.
Hello, ma'am. Good
Hmm.
evening.
Good evening. How are you?
I'm good, ma'am. How are you?
I'm also good, but my camera is not good today. My
Oh,
camera,
yeah.
my camera is not working today. Okay. Hmm. Okay. Now, so example, one of the example for this excretory organ is protonephridia or the flame cells that are the excretory structures in case of platyhelminthes. And this is the like diagram how this protonephridia looks like. I just have added this diagram for your reference so that you should know okay, how does this protonephridia looks like. No need to learn this diagram. Okay. Okay. So, and the second type of excretory organs present is nephridia. And how does this nephridia looks like? So, this is this diagram showing how does the nephridia looks like. The single unit of nephridia is known as nephridium, and many nephridium combine to form nephridia. Okay. So, these nephridia are tubular excretory structures. So, what you have to remember is you just have to remember the name of the excretory organ that is present in a particular organ, a particular animal. Suppose if it is asked, what are the excretory structures in case of earthworms and annelids, then you should know nephridia. Okay. And what are the excretory organs in case of platyhelminthes, then you should know they are protonephridia or flame cells. Okay. So what is the main function of nephridia in case of earthworm? It helps in removal of the nitrogenous waste and maintaining the fluid and ionic balance of the body. The third is Malpighian tubules. 1 minute. Okay, the third organ is Malpighian tubule. So these Malpighian tubules are excretory organs of most of the, wait, these Malpighian tubules, what happened? I don't know what is happening. So these Malpighian tubules are the excretory structures that are present in case of the insects, including cockroaches. So how does this Malpighian tubule looks like? It looks like this. It will be present at the like abdominal portion. So this Malpighian tubules helps in removal of the nitrogenous waste and also responsible for osmoregulation in case of insects. And one different thing is adrenal glands or green glands performs the excretory function in crustaceans like prawns. So these all are the excretory organs that are present in different animals, different types of excretory organs present in different types of animals. Okay. So Asmi, till now I have told them that many type of nitrogenous waste are produced in our body by different metabolic activities that are happening in our body. So these waste should be released out of our body, otherwise they will cause any harm to our body. So they may include many different things along with ammonia, urea, and uric acid. So the animals that are releasing ammonia in form of their excretory thing, or we can say in nitrogenous waste, are known as ammonotelic. Examples are given in this notes. I will be posting the notes of this chapter till today night, so you can see in that also. And the animals in which the urea is released in form of nitrogenous waste are known as ureotelic, while the animals in which the uric acid is released as a form of nitrogenous waste are known as uricotelic, out of which ammonia is the most toxic nitrogenous waste, and it requires a lot of water for its dilution, or we can
Removal.
say release. And the least toxic is uric acid. Okay, what happened, Risha?
So, ma'am,
Nothing,
humans
ma'am.
have removal of both
Urea.
ammonia and uric acid, right?
uh, Mainly that
Ammonia.
no, wait 1 minute, I've just taught you. I have given examples, like we are releasing urea mainly, urea, and we are known as ureotelic, sorry. Mammals are ureotelic, and these reptiles, birds, and snails, they are uricotelic, while aminotelic are पानी में रहने वाले water animals are mainly. Okay, so aminotelic ka examples are bony fishes, aquatic amphibians, and aquatic insects, because they are releasing ammonia and it requires a large amount of water for release of this ammonia in form of nitrogen, excretory material. Okay, so mammals are releasing urea and they are known as ureotelic. So learn all of these examples. Okay, different types of excretory organs along with the name of the animal, and different type of animals producing urea, ammonia, and uric acid, and try searching more examples on Google if you can, because this is a very common question. It will be asked, like the name of the animal is given and you have to tell that it is ureotelic, uricotelic, or aminotelic, or if it is like out of the following which one is ureotelic or aminotelic, it may be asked. Okay,
Yes, ma'am.
1 minute. Okay, so now we are going to start human excretory system. uh, Everything is clear till now? Should I proceed, or you have any doubt?
Ma'am, you can proceed.
Okay, so human excretory system, this is very interesting chapter. In humans, the excretory system of kidneys, one pair of ureters, a urinary bladder, and a urethra. So this is the structure of the human excretory system: a pair of kidney, one ureter, like a pair of ureter, urinary bladder, and urethra. So these kidneys along with this plays an important role in excretion. Okay, so how does the kidneys look like? These kidneys look like reddish brown in color, and they are bean shaped in structure, and they are situated between the levels of last thoracic and third lumbar vertebra. So what is lumbar vertebra? So this lumbar vertebra, this is the vertebra that is making vertebral columns. So the lumbar vertebra consists of 5 individual cylindrical bones: 1, 2, 3, 4, and 5. These are the individual cylindrical bones that are responsible for forming spine in the lower back, that is our backbone. These vertebrae carry all the upper body's weight while providing flexibility and movement to the trunk region. So you know, head, trunk, and what we can say, our body is also divided into three segments: head segment, this thoracic or trunk segment, and the lower below legs portion. Okay, so this is situated within the thoracic area. You know, where the lungs are present is known as thoracic region of our body. So these kidneys are situated below lungs. So they are present in the last of the thoracic cavity or thoracic segment, and the third lumbar vertebra, that is close to dorsal inner wall of the abdominal cavity, or we can simply say that the kidneys are situated just below the our like thoracic region, lungs ke thoda niche it will be situated, and like it is situated like that. It is like just above the abdominal region. Okay, so this thing you should remember. Like in case of an adult human, each kidney, we have a pair of kidney. So each kidney measures about 10 to 12 centimeter in length. It's the size of the kidney, and it is 5 to 7 centimeter in width, and its thickness is also given here. And what is the weight of the kidney is 120 to 170 grams. So these things you should learn. So you don't have to learn this diagram of lumbar vertebra. I just have added to explain you how does it look and what is lumbar vertebra. Okay, so in the center of the kidney, okay, towards the center of the inner concave surface of the kidney is a notch known as hilum. So how does this hilum looks like? So this is, the kidney is divided into two regions. The outer is known as cortex. This region is known as cortex, and inner, this region is known as medulla. This is the simplest thing to learn the structure of kidney, that the outer region is known as cortex and the inner is known as medulla. So in the center of the inner concave surface, like in center of this, there is an notch-like structure will be present, that is known as hilum, through which ureter, that is this ureter, blood vessels, these blood vessels, and the nerves enter. Okay, there is a structure known as hilum into which this ureter, blood vessels, and nerves will enter. So inner side of this hilum is a broad funnel shaped space known as renal pelvis. So where is renal pelvis? So on the inner side of this like what is this hilum, one funnel shaped structure will be present, that is known as renal pelvis, with projections known as calyxes. So these are the projections of this renal pelvis. A single is known as calyx, and they all together are known as calyxes. Okay, you all are understanding the structure, or is there any doubt? Risha, what happened today? You are not keeping well. Why your face is looking like you are sleeping? What happened?
No, ma'am, nothing.
Are you fine?
Yes,
Or the
ma'am.
class is boring?
No, I'm fine.
This is very interesting chapter, I think. अच्छा chapter है ये. Okay. Okay. So the outer layer of kidney is a tough capsule that is known as the cortex. The inside outer is cortex and the inner is medulla. Okay, so like the outer layer is this is the outer layer of kidney that is very tough, and the inner portion of the kidney is divided into two zones, the cortex and the medulla. Okay, so medulla is again divided into few conical masses known as medullary pyramids that projects into the calyxes. So these are the medullary pyramids. So this is the cortex, and this is the medulla. So again, this medulla is divided into few conical masks. They are conical in structure, known as medullary pyramids. Okay, and the cortex extend in between the medullary pyramids as a renal columns known as columns of Bertini. So what is columns of Bertini is? Columns of Bertini represents the extension of renal cortical tissues. So what are renal cortical tissues? These are the cortical tissues like named as renal cortical tissues, and they are responsible for separating these pyramids, these structures, and such are they are responsible for separating these conical or the pyramidal structures known as renal cortical what tissues, renal cortical tissues. समझ में आ रहा है ना?
Yes,
Yeah,
ma'am.
ma'am.
Asmi,
Yeah, ma'am.
Shreya,
Yes, ma'am.
is there any doubt or should I proceed?
Ma'am, you can go ahead.
मैं बहुत फास्ट चल रहा है क्या? Should I slow down my speed?
No, ma'am, it's okay.
You all are fine with that, Asmi, Anvadya, and Disha, or I am like running very fast?
No,
Ma'am, you
ma'am.
can
It's
slow
fine.
down
You
a
can-
bit.
And my speed is, I think my speed is today very fast
Yeah, ma'am.
because this is like quite interesting. तो मुझे पढ़ाने में भी I am feeling very good.
Ma'am, can you once again repeat the divisions of medulla?
Yeah, sure. Okay. So the medulla is divided into few conical masses. 1, 2, 3. You can see these are the conical masses, and they are known as medullary pyramids that projects out into the calyx. So these are the calyxes. They are projecting out through this. So the cortex extend in between the medullary pyramids. So one medullary pyramid and other medullary pyramid, the cortex that is present between them is known as renal columns, and they are known as columns of Bertini. Now it is clear. These are the medullary pyramids that are found in they are conical masses, and the space that separates these two medullary pyramid structures, they are known as columns of Bertini. And they are extensions of renal cortical tissue, or you can say a special type of tissues named as renal cortical tissues are responsible for separating two medullary pyramids, and that the area that is present in between two medullary pyramids is known as columns of Bertini. Now
So,
it is
ma'am, what
clear.
is its difference from renal cortical tissues? Cortical tissues?
No, they are made up of that only. These like
Okay, ma'am.
cortical tissues are a type of tissues that are present between these two medullary pyramids, and the area that is present between them is named as columns of Bertini, that is made up of renal cortical tissues.
Ma'am, I got it.
Fine, Disha?
Yes, ma'am.
Anvadya?
Yes, ma'am.
Okay. So, yeah. So each kidney has nearly 1 million complex tubular structures known as nephron. So nephron is the basic structure of kidney, or like many nephrons are combined to form the functional unit of excretion known as kidney. Okay, so functional unit, if it is asked out of the following which is the functional unit of kidney, then you should like select nephron. Okay, so again, each nephron, like a kidney consists of a large number of nephrons, and a single nephron consists of two parts, the glomerulus and the renal tubule. So what is the structure of each nephron? So this is the structure of a nephron. So this area, this is known as glomerulus, and this region that is present below glomerulus is known as renal, what is known as renal tubule. Okay. So a nephron is composed of two parts, glomerulus and renal tubule. So what is glomerulus? This is glomerulus, and the region that is present below this glomerulus is known as renal tubule. And these are all the different parts of this renal tubule. I will explain you further. Okay. Now it is clear what are the two different parts of a nephron. It is very important thing Try to learn it and understand it in class only. Otherwise, if you will not skip this thing, so the rest of all the portion of this chapter will not be like understood. सब समझ में नहीं आएगा if this structure of nephron will not be clear in your mind. Because these words will be repeated again and again. If you don't learn what is the like particular name of the particular region, then you will not be able to correlate the things. Okay, should I proceed, Shreya?
Yes, ma'am.
Okay, Risha, Anvadya, Asmi, should I proceed, beta?
Yes, ma'am.
Yes, ma'am.
So what is glomerulus basically? It is tuft of capillaries, or a large number of capillaries are combined to form, they are formed by the afferent and efferent arteriole. So what is difference between afferent and efferent arteriole? So they are basically responsible for supply of blood to the glomerulus of the kidney. But the afferent and efferent arterioles have different functions, or they are different from each other. An afferent arteriole is a branch of renal vein. Okay, now see the difference. They are the branch of renal vein that carries blood containing nitrogenous waste to the glomerulus, and an efferent arteriole is a branch of renal artery that carries filtered blood back to the circulation. So what is difference between artery and vein?
Ma'am, one carries pure blood, and whereas one other carries impure blood. Veins carry impure and arteries carry pure blood.
Don't use word impure and pure. Use particular.
Oxygenated and
Deoxygenated and
deoxygenated.
oxygenated.
Yeah, use like terminology. Use science terminology, beta. Oxygenated and deoxygenated. Okay. So arteries carry oxygenated blood away from the heart, while the veins are responsible for carrying the deoxygenated blood away from the tissue capillaries back to the heart. Okay. So what is difference between afferent and efferent arteriole? It is clear, beta? Clear for all?
Yes, ma'am.
Risha, Anvadya, Asmi and Shreya?
Yes, ma'am.
Yes, ma'am.
Understand them now only, because these are the basic terminologies, and the complete chapter is based on these terminologies. They will be used again and again, so don't get confused between them. Now if it is clear, then I will proceed. Otherwise you can ask me once more if there is any doubt. And one more thing, the theoretical portion for last chapter, which was left, so all of you have gone through it or not?
Yes, ma'am.
So is there any doubt in that, beta?
I don't think so.
Okay, so after once you submit your assessment, what is given, what is known, that is assessment only, I think. So submit that. After that, I will be taking test of chapter 17. Okay? Because if I'll take it now, then it will be a burden on you. So just finish that, submit your assignment. Then I will be taking test of chapter 17. So I'm telling it right now so that you can start preparing for the chapter. And I don't know when I'm going to take the test, but once I will prepare paper, then I will announce test before one day only. So start preparing that chapter from today only.
Ma'am, are you going to keep it this week?
No. You will be submitting your copies on 7th, I think. So no, not in this week. I will take test on next week. On which date I don't know. I will inform you 1 or 2 days before. But start preparing for that chapter. And if it is possible, please send me the paper pattern which your school is following, so that I can also prepare paper accordingly. Because Risha told me that school is not including 10 marks question.
No, ma'am.
Yeah,
we only for bio. I think we only had 5 markers.
And they are dividing that into 2 marks, 1 marks.
Yes, ma'am. Yeah.
You know?
Yes,
So if you have any sample question paper, or if you have any question paper, or if you remember the pattern of the question paper, please send me so that I can also prepare the paper accordingly.
Once we get our bio answer sheet, only we'll come to know the paper pattern because we don't have the question paper right now.
Okay, so when will you be getting that?
We don't know.
Don't know. And one more thing, I and sir are also like discussing about your that objective test. So we are also trying, and we will be conducting an objective test of all the chapters that are done till now very soon. Because subjective test is done, but we have to take the objective test as well. So be ready for that also. That should not be a surprise for you. So that's why I'm informing you that we are going to conduct objective test very soon. So start revision of the chapters that we have finished till now also. It's not like that we will give you a surprise. We will inform you 1 or 2 days before, but start revision at least. Okay. Okay. Now should I proceed? So first we will see the diagram of the nephron. So nephron consists of many different parts. One structure that is a bowl-like structure is known as Bowman's capsule, in which these glomerulus will be present. That is the network of capillaries, an afferent arteriole and an efferent arteriole. And this below region is known as renal region, renal tubule. This renal tubule is divided into many regions. The first portion of the renal tubule, or we can say the proximal portion of the renal tubule, is known as proximal convoluted tubule, and these all structures will have different functions. We will be discussing their functions in this chapter further, okay? And the region that is present below the proximal convoluted tubule is known as loop of Henle, that consists of descending loop of Henle and ascending loop of Henle. Okay, so first is proximal tubule, proximal convoluted tubule, followed by Henle's loop that is divided into two. The one portion that is present at the descending region is known as descending limb of Henle, and the ascending portion is known as ascending limb of loop of Henle. And this red color structure represents vasa recta. So what is this vasa recta? I will be explaining you later on. And one region that is followed by distal convoluted tubule is known as collecting duct. Okay, so these are the different portions of the nephron, and their functions I will explain you in this chapter. So the diagram of the nephron is clear?
Yes, ma'am.
So proximal convoluted tubule is, don't get confused with this diagram, okay? First will be proximal convoluted tubule, followed by loop of Henle, that is followed by distal convoluted tubule, and then collecting duct. Distal convoluted tubule se pehle this vasa recta will come, okay? So the blood from this glomerulus is carried away by an efferent arteriole, as I told you earlier also. And the renal tubule begins with a double-walled cup-shaped structure known as Bowman's capsule. So renal tubule, this is like I told you earlier also, the nephron is divided into two parts, glomerulus and renal capsule, right? Renal tubule. So this glomerulus is the first part, and the renal tubule starts from this Bowman's capsule that encloses the glomerulus, and the Bowman's capsule and the glomerulus together form a structure known as Malpighian body or renal corpuscles. Okay. So a glomerulus alone is a glomerulus, but if we consider both glomerulus and Bowman's capsule, then they are forming a structure known as Malpighian body or renal corpuscles. Remember this. Okay. So what is glomerulus? I told you earlier also. So this glomerulus is a ball of capillaries that is surrounded by a Bowman's capsule in which the urine is filtered. So the urine will come through this Bowman's capsule and will be filtered with, urine will come to this glomerulus and will be filtered within this Bowman's capsule. Okay. So the filtration that is occurring within this Bowman's capsule consists of three components. The endothelial cells of glomerular capillaries are the first. Okay. So the tubule that is followed by the Bowman's capsule is highly coiled and it is known as proximal convoluted tubule. That's why I explained you diagram first, because now I will explain you the theory. So just keep that diagram in your mind, or if you have a book, please open that. I have taken this diagram from your book only. So try to correlate the theory with this diagram. Risha, I think today you are not keeping well, beta, hai na? What happened?
I'm
Do you
fine.
have any tension?
No.
Then is there?
Ma'am,
हाँ बेटा.
I'm just reading.
Okay. Okay. Fine. Okay. So and a hairpin. The first is Bowman's capsule. That is followed by the proximal convoluted tubule, and a hairpin-shaped structure known as Henle loop is the next part to the proximal convoluted tubule. That is divided into the descending and ascending limb. Just see the diagram. This Bowman's capsule followed by proximal convoluted tubule, then a U-shaped structure known as Henle loop divided into descending limb and ascending limb, that is followed by the distal convoluted tubule and the collecting duct. So this is the same thing is written in the theory also. The ascending limb continues as another highly coiled tubular region that is known as distal convoluted tubule, and in case of many nephrons, this distal convoluted tubules open into a straight tubes known as collecting duct, and many of which converge and open into the renal pelvis through medullary pyramids in the calyxes. Okay, so this collecting duct will be opening into the renal pelvis, that is the pelvic region. Okay, through medullary pyramids of the in the calyces, that is of kidney. Okay, so that is the structure of nephron. For nephron, say how how then these nephrons are connected to the kidney, or they are present. So they will be opening into the medullary pyramids that are present in the kidney. I have told you, I have explained the structure of kidney. So these medullary pyramids, now you know where they are present in the kidney, in the medullary region of the kidney. They will be present in form of like triangular shaped structures. You remember them? Medullary pyramids.
Yes, ma'am.
Fine. Anvadya, Asmi and Risha, you all
Yes,
are able
ma'am.
to correlate the theory with diagram, beta?
Ma'am.
Yes, ma'am.
Yes, ma'am.
So it's not possible to open the diagram in each page. That's why I told you, I explained you the diagram first. Okay. So the Malpighian corpuscles, what do you mean by Malpighian corpuscles? I told you. Can anyone of you answer what is Malpighian corpuscles? I told you in before 10 minutes, 5 minutes only, I think. नहीं पता बेटा.
You told?
Yeah, I told.
I
They
think
are
there
in the
was
cortex of
something.
the kidney.
No, that Malpighian corpuscle is what? These Malpighian corpuscles are a number of small, round, deep red bodies present in the cortex of the kidney. They will be present in this. This is the glomerulus. This is the, I think, what? Okay, so Bowman's
Bowman's
capsule,
capsule.
along with, I told you earlier also, glomerulus and Bowman's capsule together are known as Malpighian corpuscles. If it is present individually, or we can say individual is a glomerulus, individual is a Bowman's capsule, and if we consider both, then they are known as collectively known as Malpighian corpuscles. Okay.
And we learned it as Malpighian tubule, so I guess
Ah.
that's why we got confused.
No, it's fine. It is also known as Malpighian corpuscle as well. Okay. And don't get confused. Okay. So कहाँ से करना था? I forgot. Okay, so these, the Malpighian capsule, that is the Bowman's capsule and glomerulus, proximal convoluted tubule, that is the first tubule that is present just after this glomerulus, and the distal convoluted tubule that is present in the last of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medullary region of the kidney. Now, the difference between the cortical and the medullary region is clear. Kidney has two portions, cortical, cortex and medulla. So, like difference between cortex and medulla is clear. पता है ना सबको where is the cortex and where is
Yes,
the
ma'am.
medulla present in the kidney, right? So, this Malpighian tubule, Malpighian tubule or Malpighian capsule that consists of Bowman's capsule and glomerulus, proximal convoluted tubule and distal convoluted tubule of the nephron will be situated in the cortical region of the kidney. So the outer region of the kidney is cortical region, whereas the loop of Henle, that is a U-shaped structure that is divided into ascending loop and descending loop, they will be present in the medulla region of the kidney. Asmi, what happened beta? Confusion हो रहा है क्या?
नहीं ma'am, I was just going through the diagram.
Okay, so this is not the diagram for this. You have to consider this diagram to understand this thing. Okay, so this glomerulus and Bowman's capsule, they will be this proximal convoluted tubule and distal convoluted tubule. These three structures will be present in the cortex, and this structure will be present in the medulla of the kidney. This loop of Henle, okay? Okay. Okay, so in case of majority of nephrons, this loop of Henle is very short and extend only very little into the medulla. Such nephrons are known as cortical nephrons. So the nephrons are again divided into two categories on basis of the length and presence of this loop of Henle. So the nephrons where this loop of Henle is very much short and will be present in very less amount in the medulla, they are known as cortical nephrons. Okay, why they are known as cortical nephrons? Because maximum portion of the nephrons will be present in the cortical region of the kidney. Okay, and a very small portion of loop of Henle will be present in the medulla. Hence, they are known as cortical nephrons. Okay. So, while in some of the nephrons, the loop of Henle is very long and it runs deep into the medulla, these nephrons are known as juxtamedullary nephrons. Okay, so how can you like how will you be able to understand the difference between them? So this is cortical nephron. You can say this is the cortical region and this is the medullary region of the kidney. So this loop of Henle will be present in very less amount in this, what we can say, what is this medullary region. So it is known as cortical nephron, and in this diagram you can see this loop of Henle will be present in the maximum amount in the medullary region of the kidney, hence it is known as juxtamedullary nephron. Okay, this is the structure of nephron. Its loop of Henle is maximum present in the medullary region. It's known as juxtamedullary nephron, and this loop of Henle will be present in very less amount with this medullary region. Hence, it is known as cortical nephron because maximum region of the nephron will be present in the cortical region. Here, maximum region of the nephron will be present in the medullary region. Okay, the difference between cortical and medullary nephron is clear.
Ma'am, does this affect like the functioning of the nephron? No, right?
No, this is just on the basis of position of this nephron. Okay? Otherwise, the functioning of each portion of nephron will remain same in all cases. Okay?
Yes, ma'am.
Okay. Okay. The efferent arteriole that emerge out from the glomerulus forms a fine capillary network around the renal tubule that is known as peritubular capillary. So please give me an answer. What is renal tubule?
Ma'am, it carries the
Ma'am,
pure
it carry.
blood. Oxygen, like the nitrogenous waste is not there in the.
Open the door. Open the door. 1 minute, huh? 1 minute. Open door. Go and sit. I'm coming. Sorry for the interruption. The efferent arteriole that emerge out from the glomerulus. Okay, so I was asking what is renal tubule. Renal tubule constitutes proximal convoluted tubule, distal convoluted tubule, loop of Henle, collecting duct. These all are the portions of the renal tubule. ठीक है, उसके पहले glomerulus will be present. Okay, so vasa recta. What is this vasa recta? That is given in this diagram. This is vasa recta. So what is this vasa recta? A minute vessel of this network, that is peritubular capillaries, runs parallel to loop of Henle, forming a U-shaped vasa recta. So this vasa recta is basically absent or is highly reduced in case of cortical nephrons. So where are the cortical nephrons? Where is the diagram of the cortical nephron? Yes. Okay. So ये है आपका, what is this loop of Henle? There should be some, this diagram it is not given, but I am trying to explain you. In case of this cortical nephron, that vasa recta will be less developed or will be very small, or in this structure, in case of medullary nephron, this vasa recta will be highly developed. This is the difference between the cortical and what medullary nephrons. Okay, understood?
Yes, ma'am.
Asmi, Anvadya, Shreya, Alisha.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
I have also added the definition for renal tubule. Okay.
Yes, ma'am.
It is
Ma'am,
the,
can I leave? It's 5 o'clock.
yeah, okay, fine, beta.
Thank you, ma'am.
Okay, bye. ठीक है? Okay, now we are going to learn urine formation. So there are three main processes are involved in the formation of urine. First is glomerular filtration, the second is reabsorption, and the third is secretion that takes place in different parts of nephron. So सबसे पहले मैं ना वो diagram ही समझाती हूँ. Filtration, reabsorption and secretion. First consider this diagram so that उसके पहले की चीज I don't know कि they are correlating or not, but this is my favorite topic, so I want to explain you. So this is the structure of a nephron. We know that this is the glomerulus, this is what glomerulus, this is Bowman's capsule, and this is renal tubule region. Okay, so there are three steps for that are involved in the mechanism of urine formation. The first is glomerular filtration, the second is tubular reabsorption, and the third is tubular secretion. So what will happen during the glomerular filtration? I will explain you. Okay. So during glomerular filtration will occur in this glomerular region. So during this, the water and the solutes that are smaller in comparison to protein are forced through the capillary walls and the pores of the glomerular capsule into the renal tubule. So through this, the blood that is coming from this, what is this, glomerulus, will be forced out of this glomerular into this Bowman's capsule. So water and smaller sized solutes will be like supplied or will be transferred from this glomerulus to this Bowman's capsule capillary walls, like through the capillary walls of this glomerulus. It will enter into the glomerular capsule, and from there it will be moving into the renal tubule. So this is the first step for mechanism of what? Urine formation. समझ में आया ना? So this is the first step. The blood that is coming with the help of afferent arterioles into this glomerulus, in that the water and small solutes will be present. They will be transferred from this glomerular capsule into this Bowman's capsule or the renal tubule. This is the first step known as glomerular filtration. Okay, should I proceed? Anuvadya beta, should I proceed?
Yes, ma'am.
Okay. The second is tubular reabsorption. During this step, water, glucose, amino acids, and the needed ions are transported out of the filtrate into the tubule. So this, what we can say, tubular reabsorption will occur in this region of the renal tubule. So these things, they are filtered out into the tubule cells and then enter the capillary blood. So this is the, we can say, what is proximal convoluted tubule, mein this thing will be happening. And these things will enter out, will go out from here and will enter into the blood that is flowing in the capillaries.
Ma'am, could you explain again what are peritubular capillaries?
Peritubular capillaries. Okay, wait. Actually, I think I should teach you this theory first. उसमें कहीं peritubular cavity this capillaries will come. But मुझे I thought कि वो diagram पहले समझाती तो शायद समझ में अच्छा आता. That's fine. Wait. I think I should start with this theory only. तभी समझ में आएगा. One minute. Till then you can see this diagram. Wait. Okay. These peritubular capillaries are the fine network of capillaries that are present all over the proximal and distal tubules, as well as in the Henle, loop of Henle also. And in the region of loop of Henle, these peritubular capillaries are known as vasa recta. So they are the network of capillaries that are present all over this renal tubule structure. So this is the structure. So 1 minute. So from here the renal tubule structure is started. So this is the proximal region, this is the distal, and this is loop of Henle. So the capillaries that are present throughout this region are known as capillary, peritubular capillaries. Okay. So
Yes, ma'am.
I think I should start with this theory only. That is okay. So urine formation involves 3 steps: glomerular filtration, then reabsorption, and then secretion. That takes place in different parts of nephron. The first step in urine formation is the filtration of the blood that is carried out by the glomerulus, and it is known as glomerular filtration. Okay. So about 1100 to 1200 ml of blood is filtered out by the kidneys per minute. Yeah, you have to remember this thing, okay? Which constitutes about 1/5 of the blood pumped out of each ventricle of the heart in a minute. So the amount of the blood that is filtered by the kidney is in 1 minute is about 1/5 of the blood that is pumped out of the each ventricle of the heart. Okay. So the glomerular capillary blood pressure causes filtration. Now what will happen? The glomerular capillary blood pressure, that is the blood capillaries that are present in the glomerular region, creates a pressure that causes the filtration of blood through 3 layers. So जो ये हमने पढ़ा था filtration process that is occurring there here. So this is the glomerulus and this is Bowman's capsule. So the blood capillaries present in this glomerulus region will create a pressure so that the blood can be moved from glomerulus to Bowman's capsule. Okay, so it will be occurring through 3 we can say layers. Okay, so the glomerular capillary, the capillaries that are present in the glomerulus, they exert a blood pressure that causes filtration of blood through 3 layers. That is the endothelium of glomerular blood vessel. This is the region of the glomerular blood vessel, endothelium region. No, this is endothelium region. This is the endothelium region of the blood vessel, glomerular blood vessels. This is the epithelium of the Bowman's capsule, and this is the basement membrane that is present between the endothelium of the glomerular blood vessel and epithelium of Bowman's capsule. अब इसको try to correlate this diagram with that diagram. So where is the Bowman's capsule? This is क्या हो रहा है? This is Bowman's capsule, right? ठीक है? This is glomerulus. So the blood from here to here can be passed with the help of 3 layers. So these 3 layers will be, first is endothelium of glomerular blood vessel. So this is glomerulus. So endothelium of the glomerular blood vessel, the layer will be like this, endothelium region. Okay, then what is the second is epithelium, that is the outer layer of Bowman's capsule. Endothelium means the inner layer of glomerulus and epithelium means the outer layer of Bowman's capsule and a basement membrane between these 2 layers. So don't learn that tough diagram, try to learn like from here only. So iski outer layer, inner layer endothelium of this Bowman's capsule, outer layer that is epithelium of this Bowman's capsule, and a layer present between these 2 is known as basement membrane. So filtration will occur through these 3 membranes. Now it is clear, or it is confusing? Risha, Asmi and Anvadya, tell me beta.
Yes, ma'am.
It is like अब समझ में आ रहा है or it is confusing?
Ma'am, समझ में आ रहा है.
Asmi?
Yeah, ma'am, I can understand.
You can understand. Anvadya, should I repeat or like it's clear beta?
It's clear, mom. You just have to go through it.
If you will go through it, then it will be like this chapter is quite easy. You will be able to understand. But I want you all to understand within class only. There should not be anything that should be like, hey, kya pada, we don't remember. So if there is any confusion, then I can repeat it. Should I proceed or should I repeat?
Proceed.
You can proceed.
Because we know that anything that is transferring or filtering is through glomerulus into Bowman's capsule. Okay, it will occur through glomerulus and go into the Bowman's capsule. So endothelium, anything that will enter into the glomerulus will go out of the glomerulus, right? So it will enter. So endothelium of the glomerulus will be sending the things into the epithelium of the Bowman's capsule, and there is a layer between them that is known as basement membrane. So filtration will be occurring. What happened? Okay. How much time is left?
15 minutes.
Okay. So the epithelial cells of the Bowman's capsule are known as podocytes, and they are arranged in a manner so that to leave some minute spaces for filtration known as filtration slits or slits pore. So this is the same thing is given in the diagram. These are the epithelium of the what is that Bowman's capsule, consists of the cells known as podocytes, and they consist of a few space between them that is known as filtration slit. Okay, this diagram is the theory related to this diagram is given here. So the blood is filtered so finely through these membranes that almost all the constituents of the plasma except proteins passes onto the lumen of the Bowman's capsule. Therefore, it is considered as ultrafiltration. So if a question is asked, like in case of humans, the filtration that is occurring through kidney or through Bowman's capsule is known as ultrafiltration. Why? Then you have to explain this structure, that the blood that is entering into the glomerulus will be filtered through three membranes, that is endothelium of glomerulus, epithelium of Bowman's capsule, and a basement membrane that is present between these two membranes. And a special type of cells will be present in the epithelium of Bowman's capsule known as podocytes. They consist of small spaces between them that is known as filtration slits, and the filtration through them occur in very fine manner. They will filter almost all the things except proteins, because the proteins are larger in size, so it is difficult for them to filter the protein. Because they are able to filter like almost all the constituents of plasma, hence this type of filtration is known as ultrafiltration. Okay, I'm sure a question can be asked on like ultrafiltration. So no one is going to draw this diagram if the question is asked other student in your class. So I want you all to draw this diagram if ultrafiltration is asked. Okay, this is the easiest diagram. You can draw it. Okay. So the amount of filtrate that is formed by kidneys per minute is known as glomerular filtration rate. So this is the definition of glomerular filtration rate. Learn it. One line question can be asked on what is glomerular filtration rate. So you should say the amount of filtrate that is formed by the kidneys per minute is known as glomerular filtration rate. So what is the range of this GFR in a healthy individual? It is approx 125 ml per minute, that is 180 liters per day. Remember this, okay? So our kidney can, like there is a mechanism, inbuilt mechanism present in our kidneys, that our kidneys can be able to regulate this glomerular filtration rate. So like a mechanism, that is responsible for maintaining this glomerular filtration rate is carried out by juxtaglomerular apparatus. This is a special type of apparatus that is inbuilt in our kidney, and it is responsible for maintaining glomerular filtration rate in our kidney. How this JG is responsible for maintaining this glomerular filtration rate? So it is a special sensitive region that is formed by the cellular modification in the distal convoluted tubule and the afferent arteriole at the location of their contact. So there is a place in the nephron where the distal convoluted tubule and the afferent arterioles are connected with each other. So when the rate of the glomerular filtration rate drops down, then the cells get modified. So this GFR can activate the JG cells. What is JG cells?
Juxtaglomerular cells.
Yeah, to release the renin. It is a hormone that can stimulate the glomerular blood flow, and therefore the glomerular filtration rate can be come back to its normal. So this is the structure of juxtaglomerular apparatus. I just have added to explain you कि क्या होता है. ये distal tubule, and this is the that is the afferent and efferent arteriole. So efferent arteriole and distal tubules are connected somewhere in the structure of the nephron. So when the glomerular filtration rate is dropped, then the cells of the distal tubule and the afferent arteriole get modified, okay, and they will release an hormone known as renin that is responsible for stimulating the glomerular blood flow. So the blood flow that will occur in this glomerular will be stimulated by the release of an hormone, and the glomerular filtration rate can come back to its normal. समझ में आया or it's confusing?
Ma'am, can you repeat what the JG cells are again?
Okay, these juxtaglomerular cells will be present in this afferent arteriole. They are the type of cells that are present within this afferent arteriole. So when the glomerular filtration rate decreases, then it can activate these cells that are present in the afferent arteriole of the nephron, and they releases an hormone known as renin that can stimulate the glomerular flow. Means the blood that will be flowing in this glomerulus will be stimulated by this renin, and the glomerular filtration rate can come back to its normal. Means the rate of the glomerular filtration can be increased. Now it is clear?
Yeah, ma'am.
Very interesting and very easy to, don't get confused. You should remember that there is a region in the, we can say, what is that, nephron, where this distal tubule and the afferent arteriole are coming in contact with each other. Now, if the glomerular filtration rate, that is the rate of filtrate that is produced by the glomerulus or kidneys per day or per minute, sorry, not per day, if it will be released, decreased, if due to some reason the glomerular filtration rate decreases, then these juxtaglomerular cells that are present in the afferent arteriole releases an hormone known as renin that is responsible for stimulating the glomerular blood flow within this glomerulus, and the glomerular filtration rate can come back to its normal, that is fixed, that will, in case of healthy individual, it will be about 180 liters per day. It should be 180 liters per day. Suppose if it is reduced because of some reason, then our kidney is able to regulate this glomerular filtration rate on its own. Okay? अब समझ में आया? अगर if there is any confusion, you can ask me. I will explain you once more. If it is clear, then only I will proceed.
Ma'am, it's clear.
Fine, Risha and Anvadya also?
Yes, ma'am.
Anvadya, you are also understanding, beta?
Yes, ma'am. Yeah.
I don't want to run. If it is clear, then only I will proceed. Okay. Now, a comparison of this volume to the filtrate formed per day, that is 180 liters per day, with that of urine released. So about 180 liters of the amount, 180 liters of filtrate will be produced by our body in 1 minute, I think. 1 day. Okay. In 1 day, our kidney filters about 180 liters of filtrate, out of which only 1.5 liters are released in form of urine. See the comparison. Our kidney is filtering 180 liters of filtrate in 1 day, but only 1.5 liters is released out in form of urine. So what we get an idea from this is that like about 99% of the filtrate has to be reabsorbed by the renal tubules, and this process is known as reabsorption. So our kidney is filtering a large amount of filtrate. Large amount of things are filtered out of our kidney in 1 day, but only 1.5 liter is released out in form of urine, that consists of the we can say nitrogenous waste. Okay, they will be released out only, and rest of all the things will be reabsorbed by the renal tubules. So distal or descending renal tubule, I think there is some area that is responsible for reabsorption, that we will be studying under this function of tubules. Which tubule is responsible for reabsorption? This distal convoluted tubule is responsible for the reabsorption of all the materials that are filtered by the kidney. Okay.
Yes, ma'am.
Okay, but I will teach till now, till this only. Otherwise, this chapter will be completed today in today's class only. It's very small chapter, so I will keep something for next class also. ये समझा देती हूँ till here. How much time is left? Tell me first.
5 minutes.
5?
Yes, ma'am.
Okay, fine. So the tubular epithelial cells in different segments of nephron, either by passive, for example, okay, it's in theory only. I will explain with this diagram. Glomerular filtration I have explained you earlier also. Now tubular reabsorption. This tubular reabsorption will occur in the distal convoluted tubule. So you know what is the difference between the proximal convoluted tubule and distal convoluted tubule?
Yes, ma'am.
Okay. So now glomerular filtration, now you understand that the amount of filtrate that comes out from this glomerulus into this Bowman's capsule, except proteins. Okay. Then everything is filtered in this glomerulus capsule. Now only a small amount of materials should go out of the body in form of urine. So the water, glucose, amino acids, and all the needed ions are transferred out of the filtrate into the tubule cells and then enter the capillary blood. So this tubular reabsorption will occur in the distal convoluted tubule region of the nephron. Okay. And third step is tubular secretion. So potassium, क्या हुआ अनवद्या को? Wait. 1 minute. I think Anvadya got disconnected.
Yes, ma'am.
Yeah. Anvadya, what happened, beta?
Ma'am, the Wi-Fi from the computer got disconnected. I don't
Okay,
know.
fine. So I think I will be stopping here only. Okay. So the third step is tubular secretion. I don't think this diagram is very good, but I will explain you the same with the help of this diagram. Okay, it's good diagram. This is glomerulus. यहाँ से the blood will come and the things will be filtrate out into this glomerulus. Things are also, the name of the things will be written here also. So this arrow represents absorption, and this arrow represents, I think, what is written here. Okay, this thing I will explain you in next class. Till then, you can only learn 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. 3 things will be occurring, and which portion of the like this nephron are doing this, I will explain you in next class. You just have to remember these 3 steps that are involved in the mechanism of urine formation. Okay. So tubular secretion involves a potassium, proton, creatinine drugs. They are removed from the peritubular blood. So peritubular capillaries are the capillaries that involves proximal convoluted and distal convoluted tubules. So the blood vessels or the capillaries that are present in this region, or they are present all over the distal, proximal and loop of Henle. And these peritubular blood are secreted by the tubule cells into the filtrate, and the things that are to be released out of the body will be entered into that collecting duct, और वहां से it will be released out. ये diagram I don't think a good diagram है ये चीज समझाने के लिए. I will be repeating this the same thing in next class with some other diagram. This is not very good diagram to explain this thing. Okay, fine. I think someone is waiting to enter. ठीक है? Is there any doubt till this?
Ma'am, you'll be sending the notes today, right?
I think I will be able to send the notes till today night. I will try my best to send you the notes. नहीं तो फिर next class में तो I have to give you only. So I will try to send you the notes of this chapter today only, because a small portion is left for me to prepare. Okay. Okay then. Bye.
Bye, ma'am.
Thank you,
Thank you, ma'am.
Bye, beta.
Thank you, ma'am.
Bye, ma'am.
Fine. Bye.